Hello, everyone, and welcome to Royal Grown Radio. I'm Michael Beck.
I'm Rick Elliott.
And we are here today at Leafworks Labs in Sonoma County, Sebastapol, California, getting down to some nitty-gritty science and understanding with Eleanor Kuntz and Charlotte Thomas, CEO and co-founder of Leafworks, research scientists, and we're going to kind of get into the basics and hopefully a little deeper on some of the pathogen, sexing, and other testing that they're doing, really looking at cannabis from a much more microscopic level, really starting to understand the plant in ways that people haven't. They're documenting cultivars and trying to raise the bar for understanding of cannabis science. Thanks for having us today.
Yeah, thanks for coming. It's so fun to have you in our little office in Sebastopol. It's always fun to have visitors. So, yeah, super stoked.
This is great. Very surprised to see Charlotte. This was unannounced and to the delight of, for sure.
Coming in for lunch and then got thrown on
roped
podcast.
in.
Let's go.
The lady who knows the most should be involved.
It's a lesson. Don't come too close to us when we have the microphones. We'll just wrangle you right in. So we've all known each other a long time from the industry. We've done Emerald Cups together and MJ Bizzes and events all over the country and work with a lot of the same clients and people, but have had limited crossover. And I'll be honest, I have a very limited understanding of what you all really do. And so this is super exciting for me to kind of get the lowdown of how you came into the business, focusing on cannabis in this way, and then exactly how the business has evolved over the years.
Yeah, the founder story in a nutshell. You know, we really started out as a botanical company, so really looking at plant identification as a whole. Because in the natural products world, when you're making any kind of product, you need to know that you're getting the plants you think you're getting. And that sounds like a very trivial thing, but once you start to get plants and they're coming to you as tiny, tiny tea cuts or powders, it becomes really hard to know, is this really the plant that I think it is? And so that's really where we started, was doing identification tests so that when you're buying these things, you can know, I'm actually getting the ginkgo I think I'm buying, and I'm not getting anything else in there.
That's so important. I never would have thought of it.
Yeah, so anytime you go to the store and on that label it has a specific plant name, there has to be testing to prove that that is the actual plant in the jar. And there are no other plants in the jar as fillers. And so just like we have food fraud within the natural product, like supplement, teas, tinctures, supplement pills, all of that, you have a lot of fraud as well. And so it's really just to mitigate that. Wow. And so that's where we started. And when we got into that, workflow and started looking at that, it became really clear that there's this glaring gap within cannabis where we kind of knew nothing. And so we really just went down that rabbit hole fast and hard and have been kind of down that rabbit hole for a really long time. So we started Leafworks in 2016, and that's when we really, really started to dig in a bigger way. And there are just a lot of basic things that we don't know collectively as scientists about cannabis that we know about other plants. And that became very, very clear to us as we started to do different things. So just if you get back to that label question, that's kind of where the rubber really met the road when we first started, where we would go to a dispensary and buy a product and it would have a name on it, and then we'd go to our friends who are growing that plant with the same name, and then we'd go to a different place and someone would give us a joint that was supposedly the same thing, and then you compare all of those things, and very rarely are they the same thing.
Absolutely. Right. It's something that's widely known in this industry,
rarely.
that everyone's got the same cut, except that they don't.
And there are variations within the way people grow things, but this is on a grander scale. This is like clear differences.
Well, this is just like the baseline, right? Like, is it a book? Is it a magazine? Is it a camera, right? Like, what thing is it? And then it's like, what kind of book is it, right? So if you say just name a plant that's circulating right now in the community that's getting some notoriety.
Runts.
Right? So it's runts. Okay, so we know we have white runts, we've got pink runts, we've got different kinds of runts. But is it runts, right? And if you test that and it's tangy, you're like, okay, not the same thing. So it's really just, what is that plant? And then you can start to ask those other questions, which you're really alluding to, which is, if you grow it and I grow it, yours might be fire and mine might not be so great, right? Or there may be different characteristics, and that can do with how you're growing it, with style, in the ground, hydroponic, whatever. Or it could be the environment itself. Is it really cold at night? Do you have really hot, all of those environmental conditions.
And that's one thing with the cannabis plant that is really responsive to those things, probably more than a lot of botanicals, I would think, right?
It is very responsive to its environment. So in plant talk, it's called plasticity.
Okay.
Cannabis is very phenotypically plastic. So if you put it in one place or you put it in another, the outcome can be very different. So it has plasticity, even though the genetics are identical. So you could take a clone, so the genetics are 100% the same. It will express differently in those different environments. A lot of our medicinal plants are like that, which is why we have different qualities of plants. And so you also see it in chilies. Hatch chilies are worth a lot more. When you grow the same chilies down in my yard, here in Sonoma County, they're not going to be that spicy and delicious because my environment isn't pushing them to be that way.
Gotcha.
And so it happens in a lot of the plants where we enjoy the secondary metabolites. The environment really can play with those in a bigger way.
Interesting.
Yeah.
It's got to be incredible coming from a science background, right? Being scientists and being able to dive into virtually a new avenue of science, right? Because obviously with botanicals and other plants, there has been a lot of research done. The market and the industry has been there to support that for many, many decades, if not longer. Where with cannabis, this is like a brand new avenue where it's got to be exciting coming from the scientific mind where you're breaking through things that may have never been discovered before, right?
Yeah, it's super exciting. And there's so much low-hanging fruit in the genetic arena where we don't understand the genes that are driving flowering time or any basic things that you know these things about wheat, you know them about corn, you know them about all of the the major agricultural commodities. That very well how those things are manipulated by the differences in their genetics. But cannabis is just a wide open place. So it's super fun to dive in, and that's sort of why we've stayed down that path for so long, because there is so much to learn. There's lifetimes of things to learn.
With botanicals and other plants, I know we've all experienced it with cannabis, where obviously the market drives a certain variety, whatever's hot for the season. And then there's been this long known thing where people are just calling it whatever's hot, even though they are well aware that it's not. Is that something that has also had a place in traditional plants and nurseries where there's a difference in genetics, where the same kind of testing is there to actually validify that stuff?
Yes, and there are certain crops in which it's used more routinely. So hops is a really good example.
Okay.
One kind of hops is not worth the same amount as another kind of hops. And so if you're buying, a very like a mosaic hops or something that has a really either trendy appeal right now or a very specific and powerful profile, smell profile, that'll be worth 2 to 3 times as much as maybe a Cascade hop, some more readily available
so that's
hops.
super easy to fraud if people aren't aware.
Yep, especially how you sell the product, right? So with hops, you're not buying them from the vine. You're buying them in pellets, generally speaking, to put in your beer preparation. So you can imagine you can put all kinds of other stuff in that pellet. So it can either not be the right variety, or it can be the right variety cut with something else.
Right.
Or, so there's all different kinds of ways that kind of fraud happens. But this type of testing where you're looking at the genetics and you're looking at, is it really mosaic, is fundamental in those industries. The same thing happens with roses.
Of course.
The same thing happens with grapes, right? So I think there was a really funny example when we first moved to Sonoma County. We were talking to the ag commissioner, and he's like, yeah, there's, I can't remember the number, so I'm making them up, but there's, 100 acres of Chardonnay grown here. And even at max capacity, those 100 acres can only make, 100 pounds or whatever it is. And we have 4 times that many Chardonnay grapes being sold in the county this season. So there's fraud because there's not, the numbers don't add up, right? And so there's fraud in all kinds of things where you're either disassociated with your supply, like you're not the grower growing those plants that are going into your product, whether it's wine or cannabis products or beer. So it happens a lot.
How do people, and how do you, in the testing mode, establish what is the true runts cut? Because there's probably 10 people that are like, no, this is the runts.
Yeah.
So which one is it? Where do we start that? Who's the breeder? This is the original seed. This is where the cuts originated in the breeding program. came from? How do we establish that, and how do you back check it?
Yeah, it would be lovely if there was an easy answer to that question.
It seems difficult.
It's very difficult.
Going forward, I would imagine it might be easier as you're breaking through with this research, where when somebody is coming up with a cut, they can actually make that stance and that claim right then and there, right? So then going forward, you've got that data and it's backed up.
Yeah, and so we're kind of playing catch up, right? We have a whole bunch of different things, and they are different. Like anyone who's grown these plants, now there's distinct and drastic differences between them. So it's not like they're all the same, which some people like to say, which is like, no, you just don't know anything about this plant if you think that.
Right.
But it's really codifying the names. And it's not my job to do that. And especially being a genetics company, it's not our job to do that. It is a job of the community and the growers that have been working with those plants to do that. And so it's definitely a give and take. So the way we approach it is many times we're very fortunate to have direct interactions with the original breeders. And so that's a very good way to say,
That's a good
this
starting
is
point.
the thing that is called X, Y, or Z. And this is what it looks like. These are the characteristics. And then we can take the genetics and make a genotype of that plant. That's much easier if it's a clone, right? It's a clone, it's the same. If it's a seed plant, you have to understand that there's variation within that. And so we have to capture that whole sort of population of seeds to understand, and also understand from the breeder's point of view, where are they going with that seed, right? Like, does it have to have certain smell characteristics? Does it have to be short? Does it have? It doesn't actually matter, right? I think that's what we forget. Breeding. It's like, I think it's easier to think about it with dogs, because not all German Shepherds are clones.
Right.
They are related. They've got different moms, but they all have certain characteristics that holds them together, so that when you look at them, you're like, that's a German Shepherd.
It's a breed standard.
Exactly. And so that's what we need with our plants, with the cultivars. With the clone, it's always the same. It's the same as the same. But then when you have the seeds, it's much more like a breed standard for a dog, and that concept of they are all genetically related, but there's variation there. But what are the characteristics that are important? And so in plants, you might have cultivar characteristics that hold them together, but you don't actually care if the flower is pink or white, right?
Okay.
It's still the certain kind of daisy or whatever it is, or a certain kind of rose.
Right.
But then in other instances, the color of the flower actually matters. So it just matters how you define it to make that name. And so we really start as much as possible with people who are doing that work themselves, doing the breeding. And then in other cases, you have people who've held plants forever, and so we intake them as personal collections, and it's associated with that collector or that organization, that farm. And then the really nice thing is if we get 6 or 7 of the same thing, you can then compare them and say, oh, out of these 6, these 5 are the same, but this one is not. So you can kind of weed the one that's not out, because the collective has kind of spoken that this is what this name means.
Makes sense.
And so we approach it in both ways, and it's an iterative, very community-driven process where we hold a lot of that information, but aren't the ultimate decision makers, if that makes sense. It's definitely a collaborative way of doing it, and one of the ways that we help people who've bred those plants document those plants is we make herbarium pressings.
So what is an herbarium pressing?
Yeah.
Actually, we could show you one.
Yeah. I was saying, Charlotte is sitting next to some right now.
Yeah, let's see. Okay, to be very careful. These are some plants from Hogwash Farms, shout out.
Shout out to Hogwash. That's awesome.
Ooh, they smell delicious. Yeah, so basically you take a little cut of the plant and you press it. And so you're putting a lot of pressure on it. You let it dry out for a couple of months. And this is what it looks like. And then so what you'll do is apply some special glue, apply it to a special paper, and then this acts as a reference material for hundreds and thousands of years even.
Yeah, there's herbarium specimens that are hundreds of years old. So
Yeah.
in the My family is from Sweden, and it's very fun. You can go to the Swedish Natural History Museum in Stockholm, and they have the original pressings from Linnaeus of Cannabis sativa.
Wow.
So, and that's hundreds of years old at this point. And so it's fun, and it's nice because we take these, this is a very late flower pressing, and so it can be hard to capture the beauty of the plant at that late stage. We'll also take flower pressings a little bit earlier on, and you can really see the flower a little bit more clearly when it's not quite so mature.
Yeah.
And then also we'll take vegetative pressing, so you can really see how's the structure of the plant when it's in that vegetative state, leaf shape, internodal distance, all those things that kind of make a plant a unique individual.
Yep.
And so we document it this way. This is the most basic way to establish IP, right?
Okay.
There's a lot of talk about sequence your plant and you own it and all this. It's kind of bullshit. Actually, it's not kind of bullshit. It is bullshit.
Right? How do you claim ownership of those things?
Well, also, any of us can walk into a dispensary, right? And then bring it to a lab and sequence it. Does that mean you own that plant?
Right.
No. I mean, if I go into the grocery store and sequence a gala apple,
You still don't
I have
own
a
gala
sequence
apples.
of gala apple, but it doesn't mean I have any ownership over that plant. And so that idea that just because you sequence something, you have ownership is very frustrating because it is totally inaccurate.
I've seen varying platforms kind of leaning into that concept, and I love that people are trying to find a way that you can, hey, I've held this seed for 30 years, I found it in a bag, but still, I mean, you didn't breed that bag, you got it from some random person in some random place, and like, do you actually have ownership now of that genetic? That it does get tricky.
Well, it's a weird world, I also think it's very funny because I think at least in this community, we like to talk about owning cannabis plants, and then if you talk about owning like lavender, people are like, oh, you can't own life, and you're like, okay.
Right?
It's a little bit of a disconnect there. And so it's really, do we own plants at all? I mean, I could go down that esoteric philosophical wormhole. But there is something if you've done work. And I think that's the important thing is in order to gain intellectual property protection over a plant, you really need to show that you've done something. So that's one of the reasons why the vouchers are so important. It's prior art. So it's an immutable object. It's not sequence. It's like an actual thing that you can then reference, and you can take sequence data from and attach it. Say this sequence data came from this thing. We put it in the herbarium on this date, so you have some time reference of actual material. And then if you continue breeding with that plant, and 3 or 4 generations down, you've really changed its chemical profile, or you've really changed its shape. So now it's much, instead of being a 12-foot tall tree, which is impossible to grow, you've bred it so it's more contained in size. Whatever you've done. But this is one of the ways you can document work.
Okay.
Right? You've done work, and this day it looked like this, and the genotype was this, and 4 breeding cycles down it looked like this, and this is how the genotype has changed. And if you have seeds, hopefully, as you get, the diversity gets narrower and narrower as you get towards what you're, going for. You're weeding out the things that aren't contributing to that phenotype. And so this is just another tool to show you've done something. Yeah. So that then maybe you deserve to have protection over that thing that you've created.
So correct me if I'm wrong, but I'm thinking the easiest example that I'm identifying with in my mind is say something like the freak show.
Oh my God, that's exactly what I was thinking.
Because it's an easy example, right? It's a clear difference from what everybody notices as a cannabis leaf. So is that an accurate example? Is that, and then they're breeding it down and they like maybe not claim ownership, but propriety over that.
Yes, and I think freak show is a really interesting example because you can get to that. Like I've seen 2 different freak shows that are coming from different places, and they're not necessarily genetically related, but they have the same mutations that make the leaves look like that. Right?
Okay.
And so it's really similar to something like in the plant world, like redbud. Those trees that are bright pink, and they before they leaf out, they're
all
super
over the East
gorgeous.
Coast and Southeast,
they're everywhere. And you have varieties that are alba, redbud alba.
Okay.
And that variety is a white variety, but you can make a white redbud in many different ways. Right? You just have to break the gene or the gene something in the pathway that makes the flowers turn pink. So it's sort of similar where they're not exactly identical, but they have the same phenotype, and so they're lumped in the same category.
Wow.
It's so it's a weird world when you come. So that's the thing about genetics, right? Genetics are supportive of IP, but they don't drive the IP. Does that make sense?
That does make sense.
It's like an added tool.
And this is a really enormous concept to wrap my head around as somebody who's not living in the genetic world like that. I mean, it's really kind of perplexing to me to see how these breeders are all doing similar work and collaborations and independent work, but actually coming up with something unique that could be labeled as individual intellectual property. Like, so much of it isn't maybe going to cross that threshold to being.
Yeah, and even if it's not intellectual property, just because we don't have an intellectual property over a German Shepherd, you still, when you're like, I decided that that's the kind of dog I want, and then you buy this dog and it's a pug, like, my daughter may be stoked, but if I'm expecting a German Shepherd, I'm going to feel irritated. And so it's really just about also being able to communicate with each other about what we're talking about.
And authenticity.
Yeah, so it's not just about I own this plant. It's also having a common language so that we can engage in commerce in such a way, or even not just commerce, just in, interacting, so that we're talking about the same thing.
Right.
And so that's why it's really important, and that's why we've really dedicated so much time to this work, because it's this humongous problem. And it's so complicated that it won't be, we can't solve it all. So we're just contributing in the way we can and offering that out to the.
Do you see that, obviously the research and the data that you're collecting now is based on what's already out there, right?
Mm-hmm.
You're trying to understand what's already been put out there. Do you see at some point in the future where your research and the information that you're collecting now is available and would be put to use for growers and cultivators to actually put together new strains with some of the better characteristics that they're looking for? Is that something that you could kind of go back and actually offer that as a service down the road?
So we're already working on doing that, and we do that type of work with various groups that are doing breeding. So we don't do any breeding ourselves.
Yep.
We interface with other groups that have plants and populations of plants that they're breeding. And we can help move different cannabinoid profiles forward. We've done a really deep dive in rosin production.
Yeah.
Sorry, I'm blanking.
My brain is going there.
Yeah, so we were looking at different things that contribute to yield within, plants that are good squishers, good at different types of extraction. We've looked, which is super
that's
fun.
amazing.
And
I wondered
it's like
about the DNA
research.
breakdown, like being able to look at it from your eyes with your expertise, if you can actually pinpoint that, similar to how a grower or a cultivator would be able to look at that from the actual flower and the way that it grows itself, right? That's generally how we're putting strains together, as you pick out the best one, the one that's got the characteristics that you're looking for in your grow and the smell and the taste and all that.
But they're looking at data.
And
not like
well, it's super complementary. Those two things go together. So
Yeah.
first you have to have the grower doing that work.
Experiential knowledge is crucial.
It's fundamental. You can't do the work we do without it.
Yeah.
So then what you do is you take that fundamental experiential, tactile, visual interaction with the plant, and you group them into categories that say, let's just say squishing. Really good at squishing, doesn't make any, squish it, nothing will come out, right? So you have to look at those categories, and then you find, every time you have something that doesn't produce any rosin, it has these markers, or it has these variations of the gene. And every time it's really, really good, you have a combination of these genes. So then you chase the genes that are positive, and you eliminate the genes that are not yielding the thing you're looking for. And so what we offer is, once you marry those two, like that visual with that bioinformatic work, then what you can do is say, I'm a grower and I have room for 1,000 plants. But you can germinate 10,000 plants and then screen them for the 1,000 that are most likely to have the things you're interested in. So it just speeds up
That's a huge
the
leap forward.
process.
Yeah.
And it's iterative, right? So then if you do that and you say, this one still really wasn't very good, we look at that again and say, okay, what's in there that we missed so that the next time you do it, it's like sharpening a knife, right? Every time you hone in on those positive and negative attributes. It's really fun.
That's so amazing. I can't tell if
teamwork.
it's the latte or I'm just getting excited. This is the sort of thing that like just gets my brain turning, and looking and seeing like the stacks of the herbariums here. Like, so anybody can reach out to you guys online and be like, hey, I'm doing this work and I'd like better data. And they can send you samples, plant, and you can provide this as a service to any breeder anywhere, essentially.
Yeah, so we work together to make a game plan. What do you have? What are you looking for? And then we make a plan on how to work towards that together.
That's great. And this isn't something that's obviously just California based now with all states going legal. This is something and I can't imagine that there's a lot of folks doing the work that you're doing in other states at this point.
There are very few of us, just in overall.
Yeah, globally, I would assume there's not a ton of people.
Yeah, what comes to mind is at MJ Biz when we ran into each other. You came over to the booth at some point, and I had been talking to an old friend of ours, one of my old neighbors from my old grow, Duke at Royal Bloodline.
Yeah. Okay.
And then we were talking, and I introduced you, and then I got kind of tied up and pulled away. And I just remember, I love Duke, and he's super knowledgeable. He's a great grower. And I just remember you had to go, and then he's still standing there, kind of looking just like at this grin on, and I'm just like, man, it's good to see you, buddy. And he was just like, man, thank you so much for introducing me to her, because she's fantastic.
No, that's awesome.
And so this conversation I've been very, very excited to have for a while. I feel like this is one of those episodes where I might be the co-host and part of the conversation real time. But I'm like everybody else that's going to be listening to it with open ears, like, holy shit, this is incredible.
Yeah, I find the best episodes are the ones where we're the audience and like, no, I'm actually interested. What's going on here? And all this ties together for all of the knowledge that's going into terpene identification, volatile sulfur compounds, understanding that esters and phenols and the contributing factors to the odor and the effect, right? So this is base science that plays a role in every level of kind of elevating traditional cannabis breeding right now.
Absolutely. And I think it's really fun because all of those secondary metabolites, whether they're the esters or the flavonoids, they also have these roles for the plant, right? Whether it's protection or pathogen resistance, or they interact with the environment too. And so we're learning not only about how the genetics influence what is possible to be produced, but also how you take that potential and then work with your environment to either elevate or suppress that potential based on what you need. And so that's like the most exciting interface for me personally, is how do you take that genetic map that exists within an organism and understand how to make it grow to its best potential. So that's a super fun magic interaction between genetics and the world that we're in. So it's so fun.
This isn't genetic modification. This is an acceleration of like Mendelian genetics.
Yeah.
Okay.
Yeah.
I think that's important because I think a lot of people, there's all this talk all over now about GMO cannabis and you're probably smoking it now, especially in major cities. And I don't know that there's science to support that. And there's a lot of talk about it, but I think that it really lends credence to the fact that most people are not going that route through genetic modification at this point. It is Mendelian style genetics, traditional breeding, but with an elevated knowledge of what you're trying to achieve.
Yeah, it's just streamlined. And the same tools can be used to do GMO breeding. Like you have to understand the genes to be able to modify them, right? So understanding the genetics is the primary first step. But marker-assisted breeding, which is what we're doing, is really just looking at what's in there and how do we pick out those things that the plant is naturally doing, and or to speak to your point, say, I know that let's just drought resistance, right? Like I live somewhere where it doesn't rain, so I need something that's drought resistant. And I can say, oh, I know that this plant that I'm working with has some characteristics that are drought resistant, but what other plants can I breed with to increase that, right? So it's another tool of screening, okay, who are my potential mating partners for my breeding population to move it in the direction I want. And so you can use it in all kinds of ways to sort of screen what you're going to do.
Right, and for different ways to create that drought resistance, whether it's, okay, this genetic loses less moisture to the atmosphere. This pulls more from its root zone. Like, there's different ways then to express those characteristics. And
100%. Like, your leaves can be smaller or your roots can be deeper, right? Like, that's two ways to get to the same outcome.
Right.
And you can kind of chase a lot of them at the same time, which is fun.
That's fantastic.
Yeah, it's
So
interesting.
fascinating.
So you're doing a lot of the research science. Like, how are you looking at these things?
Lab manager as well, right?
Yes.
Charlotte is in charge of the ship.
Well, you know.
Yeah.
That's a pretty big task.
Yeah, how do you look at this data? Are you like then amalgamating the data, or you're just trying to get it, categorize it for each piece, and then put it in an accessible drive sort of
Yeah, so I'm not actually the one that's looking at the data. That's our bioinformatician, Jeff Preet. Shout out to you.
Okay.
I'm more of the one intaking the samples, making sure that they're put into our database the right way, and then extracting DNA or RNA or whatever it is that we need from it, and then getting those samples prepared to either be sequenced or run on the qPCR or whatever we're going to be doing with them. And so that's kind of more my side of things.
Yeah,
So-
she's in charge of all the testing. So if you send us a sample to sex test or any of our pathogen tests or any of our cultivar, like if you're registering cultivar, Charlotte is the one who will do all of that, orchestrate all of that work.
and make sure that data is clean, accurate, correct, and
Yep.
categorized.
Exactly. Yeah,
Wow.
I don't know if there's enough time on this episode to really talk about everything we do, but you know, we did talk about the consulting and the herbarium work, but then we also have all of our bread and butter tests that are coming in daily and hundreds of samples a week, which is our pathogen testing and our sex testing. And those two tests mainly, we're getting hundreds of samples a week. So
Amazing.
those are kind of daily tests that we have, but then we also have our fun research side projects that come in where we're, you know, doing deeper work. So
Well, I'm glad you touched on that, because pathogen testing is all the hype right now, because everyone is dealing with pathogens, whether they know it or not. Whether
Yeah.
you think you have pathogens or don't, if you haven't tested, you most likely have it somewhere and need to start looking at this from a bigger picture standpoint. And there's not a whole lot of places that can accurately and effectively categorize these pathogens, and that's something we run into all the time. Being at a soil company, people call us and they're like, we're having this issue, can you help? Mm-hmm. And we're limited only, okay, well, we can look at the soil, it didn't start here. Well, where do you go for that information?
Yeah, I mean, pathogen testing is an interesting beast, because it's definitely increasing. We've seen a lot of people are starting to really attempt to get ahead of their problems, which is lovely, because in the last couple of years, it's mostly been triage, like, oh shit, something's happened, and it's mostly just knowledge what has happened, which is great. Mm-hmm. But this season, it seems like we're starting to get more individuals and organizations that are like, okay, we want to take stock of what's going on before we see something.
Yeah.
And so that's really where we've moved in our interface with organizations as much as possible, as much as people are willing, or to instigate or to start like pre-testing, so that we can get ahead of problems and mitigate the the negative effects of what's going on. So we're really looking at like programs, like testing programs. We're going to test this if you know, depending on if you're nursery or you're intaking things from outside, like whatever your particular situation is, how do you deal with those things so you don't bring pests and pathogens in?
Yeah, and
Is really where we're trying to focus, because
gotta
that's
be
the most
hugely
helpful.
difficult.
Yeah.
But it's hard, and I think Charlotte can speak to this. It's kind of like us. If you've got cancer and I take a cell from here, I might not have cancer, but if you take a cell from here, you might see the cancer. So because pathogens move through the body of the plant, you have to do a regular testing program sometimes to catch them. Because you imagine when a pathogen first enters, there's like 1 or 2 molecules, whether it's a virus or whatever it bacteria, it doesn't matter. There's like very few of them in the plant.
Before it starts replicating.
Right, and so you have to actually catch it where it is, and that's a lottery if you're going to get where you find it. So, but if you test frequently enough, then you can start to say, oh shit, we see it in this tray, or we see it in this mom, and then you can prevent it from spreading. But if you just test things once, and especially when they're nice and fresh and young and new, they may be infected, but you can't catch the infection. And so I think that's one of the challenges that we're having as a testing company, is really articulating the diligence that you need to have in order to catch some of these things, because they are moving targets. They're also living organisms that are breeding and replicating within the system. And by the time the plant looks sick, it's pretty easy to find what's going on, because there's so many of the molecules in the plant that you can. So that's really the challenge. And I think you can speak to that a little bit too.
We've even found, we've taken punches from one leaf, different sides, and found HLV on one side of the leaf and didn't find it in the other. And it's just crazy. So, we've iterated upon our tests multiple times to always make it better. As the research comes out, we'll change how we're taking samples, or we're constantly making it better, because it's so important about how you're taking the sample and when. And I think when we first started doing this, a lot of customers thought, or they didn't want to test as much up front, or they didn't want to do a program, where you're testing, because, maybe they didn't want to spend the money or whatever it was. But now I think things are changing. And we even had when we first started doing pathogen testing, sex testing was more of our main test. Every day that was what we were doing. But now I think it's kind of shifted. And we used to have such a high busy season of sex testing, and now it's just constantly pathogen all year round, because it's such an issue.
Given that HLV has been around in the hops industry for as long as it's been, are you able to lean on any of the data and protocols that have come out of that industry? And does it translate? I know that the plants themselves are very similar. Is it that common? Yeah,
and if you look at how a hops latent viroid moves within hops, it's very similar to cannabis. So that's another one of the challenges, is you can have infection. So it can be coming in through a leaf, but that viroid is moving into the roots first. And so you can do leaf punches all day and it's going to come back clean, because it's in the roots. So that's why we are very adamant that if you really want to test, you need to test the roots. Okay, that's not always possible. If you just cut 100 clones from moms, they don't got roots to test. Right?
Yeah.
And so it's always time and place appropriate, which is why we have these programs, or like, in this situation, this is how we suggest you test, right? They're just suggestions. But it really looks at the biology of the plant to get you the answer you need, which is what's going on.
I'm sure it's such a huge challenge when you're looking at sample size versus scale. Somebody's got 18,000 plants that are 6 feet tall. How much can they effectively send you as a sample? So that's got to be really difficult.
It has to be cost effective. You can't have a test cost $25 if you're selling the clone for $1, right? Like, you have to be able to meet somewhere. And so that's why we've really focused on batch testing. Like, how can we test multiple plants at once so that we can start to get at some answers for you? We have a testing program where we really look at moms and then move forward into the clones. So you can really start at the base and you do more robust testing of the moms, and then follow the clones to make sure nothing has happened along the way.
Okay.
But it's risk mitigation, right? You're doing what you can to catch it. And so you style the testing based on the situation you're trying to commit to. And then there's also like the annoying things where people are coming to you because they want a clean test. They don't actually care if the plants are clean.
They just want a clean test.
They just want a clean test.
They want the bill of health to be able to take to market or what have you.
To sell on. And so that's also very challenging, because,
that's not your goal.
Well, and also it's not our goal at all.
No.
But it's a different interaction.
Well, and you see that with all of the THC testing right now.
Yeah, and it's not really what we're about. We're really here to help you solve a problem and fix a situation, not so much give you a pass to be able to sell something on that maybe shouldn't be sold on. Yeah. Well, so it's a little challenging at times.
Yeah, it seems like the goal is largely to accelerate the universal knowledge of the cannabis plant and the other herb plants that you're dealing with as well. And, you know, it seems like the business is being captured by the demand for cannabis testing in one way. I'm sure you do other things still as well, but it makes sense with such a void of this type of information that people are going to be flocking to you more than ever in the coming years.
Yeah, and also expanding the types of pathogens that we're testing for. Like we have a lot of really amazing friends in the industry that, you know, this weird thing's happening, and they give us their weird, you know, same thing that you experience all the time. This weird thing's happening. Can you help me explain it? And so that's been really nice because we have a very deep root system into figuring out what's going on the ground. And realizing what's going on in Michigan with our Michigan farmers is very different than what's going on with our Northern California farmers. And then some things are kind of like HLV is pretty universal, right?
Yeah.
Like we've got people out of country that send us stuff to test for HLV. So it's sort of a global issue at the moment. But some of the fungal pathogens are much more regionally problematic. And so we really just try to dig in. And when there's new things, you know, gauge, is it a personal problem or is it a more universal problem that we should make a test to help more people with?
That's really interesting. I mean, I guess it makes sense that it's regional. A lot of these things are communicable diseases, essentially. So you see something break out in one corner and then spread, and I bet it would be really interesting to kind of map the way some of these things have moved nationwide. Like, oh, it started here, then moved to Northern California, and then moved to everywhere from there.
Yeah.
Or,
Yeah,
you
can you
know,
imagine
That's generally been it, is the Northern California
And that
explosion,
one.
because the cultivation has always been in such
Yeah.
a mass scale over here.
Things are moving
Yeah.
all around all the time. And we also read a paper where it was if you touch weed, you grind it up with your fingers, you can actually still spread HLV because it is such a small, it's an
Stable.
RNA viroid, an RNA-based viroid. It's this tiny, stable molecule. And so it's really hard to break open and get rid of it. And so it's just crazy.
Yeah, so imagine you get some weed from your friend and you grind it up and then you go touch your plants and you infect your plants. It's like terrifying.
Which, who isn't doing that, you know? Like,
well, yeah, and we go and tour a ton of farms for this, and we're always really careful. We change clothes, change shoes, disinfect, all of those things. But
you just blew his mind. I mean, I watched at least an hour a day of him rolling joints and breaking up weed in his fingers. So
yeah, well, it's pretty crazy too, because that's not something people are concerned about. They want you to take a shower, scrub your eyeballs, disinfect your shoes, but then they'll be like, "Oh, here, roll this. I just got this weed from Billy. Roll it up real quick, and let's go take a tour."
Yeah.
Yep.
So wow, that really did blow my mind.
That's
I
wild.
I know we read that and we were like, oh my God.
We collectively were all just like, what?
Yeah.
In trouble.
Yeah.
Damn you, HLV.
Yeah.
Right. I mean, it's always been understood just as far as like your scissors and all that stuff, as far as cutting clones, just cleanliness.
Yeah.
I've always just thought, God, like it would have been cool if the hops industry, had given us a heads up like 15 years ago. Like, hey, you guys are probably going to deal with this at some point.
Yeah.
Well, and there were people like, other scientists and advanced thinkers 15 years ago talking about that. They're like, just wait for it. It's out there and it's happening. We just don't have a way to categorize it or catch it yet.
Yeah.
And I think it's been really, about 5 years since it really exploded into the top of people's minds when they're losing crops and, actually dealing with the losses that force you to deal with the problem.
Totally.
And that's been like that with everything, especially in Northern California. I mean, as a cultivator for 30 years, it's like, the spider mites, and all of a sudden it was just like, oh my God, everyone's farming mites now. And then you get russet mites and everything. It's like every time you get something, you're like, what do you have? Never heard of it. And then all of a sudden, everybody has it.
Powdery mildew is the classic example of
Big
that.
time.
Ugh.
And that's there's always been like the conspiracy theorists, they're like, they're coming to get us, they're dumping it from helicopters.
Yeah.
There's always that.
Yeah.
Well, and I would think that long term, the knowledge you're capturing can help kind of negate some of that baloney in the industry. You're like, actually, no, we've seen this in data sets coming from this place and this place and this place and the different variants. So,
I mean, I dream of a day when we have a CDC, but for cannabis. or for plants in general. So it's kind of like what our ag research stations should be, but they don't really talk to each other very well.
Yeah.
Like, oh, we're seeing this disease here, and we're seeing it in these crops, and this transmission, and oh, we have a soil vector, we have a water issue. You know, just getting all that information in a place so we can understand how some of these things track. Like, we understand how we pass, you know, human germs so well because we have a central repository for the information. We know where the outbreaks happen. We know the numbers. You can also sequence those boogers and see how they've changed, you know, like how they're related to one another.
Right.
And, you know, that kind of thing could happen, but it's a lot of teamwork. So that's the issue, I think, with us, is that not all of us are very good at teamwork.
That's important.
And it takes a collective really to do that too. So
we were talking earlier about kind of the differences in traditional ag where it's all mapped out for wheat. You know what you need to feed per acre, you know what your expressions are. Are there better baselines for tracking these pathogens through traditional ag? Are they like, oh hey, this variety of apple is going to get fusarium and it's contracted this way? Is that knowledge more advanced in traditional ag?
It's so much more advanced.
Okay.
Yeah, so my family is a farming family from Ohio, my father's side. And, you know, if you're going to grow broccoli in Ohio, it's a totally different cultivar than if you're going to grow broccoli in Georgia, right? It's like you would not grow the same plants at all. They won't die. They won't live in the other environment. And so we'll get there eventually with cannabis too. Like, oh, you're in the Northeast. These are the types of things that do really well here, or these are the things you're going to struggle with, you know? And that's going to happen.
Yeah. And you see the experiential selection happening like that already.
Absolutely.
SoCal varieties are different than NorCal varieties than Michigan varieties where I grew up. And, you know, you're dealing with an environment where it rains all through the summer versus California where we get not a drop for 4 to 6 months depending on where you are. So
Zone-specific cannabis strains?
Yeah, and I mean it's complicated because it's the environment choosing, and then it's also the culture, the people choosing. So it's kind of double. But I see a future where that's 100% going to happen.
Yeah,
and I think it's going to get even more diverse because I don't know. In this sort of zone, we tend to only think about cannabis as ingestible cannabis, you know, like.
Right.
But there's so many other kinds of cannabis, you know. We like to call it hemp, but it doesn't matter if it has THC or not. Like some cannabis plants are really good at making fiber, and others are really good at remediating the soil, and others are really, you know, there's so many utility cases that are good for building materials and, you know, hempcrete or, you know, but it's cannabis.
Absolutely.
It's all cannabis. And so, you know, we may find like, oh, if I, you know, I was in Georgia for a really long time. Maybe it's not a really good place to grow consumable cannabis, but it's a really good place to grow textile cannabis. You know, I think there'll be regions like that as well, and we're just there's so much to learn that we're not there yet.
Yeah, it's in its
as
infancy.
a collective.
And obviously cannabis has been around long enough that it's probably got a genetic that fit almost everywhere on Earth, right?
Yes.
I would think, just about.
Yeah, it's one of our medicinal plants. It's like, I always joke, like, the house is burning down. It's like not a very funny joke living around here because it's like a little too close to home, but you know, what do you put in the car? You put your family, which is your pets. You put your medicines, and you put your food.
Yeah.
You know, where you travel, what do you take with you? And so, like, cannabis is in that category. It's been a food and a medicine, and
for millennia.
Yeah. As long as we've been moving around the planet, probably.
Wow.
So it makes sense why it's really found its niche in all these different places, because we've caretaken it and taken it everywhere. It's an important plant to us culturally as humans.
So
I think we forget that.
How can people actually get engaged with you to get going? I'm sure there's going to be listeners at home who are like, oh my God, this is everything I've been thinking about and want to get involved with, and this lab I'm working with can't give me that data the way I'm asking for it. And because it does seem like you're taking a different view on science, but how do people get involved?
Well, a lot of ways. I mean, they could reach out through our website mainly. Yeah, I guess it's just depending on what you want to get involved with and having that initial conversation so that we really understand what you're trying to do. And but if anyone wants to get involved with our herbarium, working with farms to do that and accept new samples. And the herbarium is my favorite part of my job personally, and I think of Leafworks as well. I think we are making history with this, and I think one day it will be so important. I bet you there will be a museum. And Dr. Eleanor Kuntz will be painted on the wall.
The plants will be on the wall.
Yeah. But yeah, I just think it's really important for the community to get involved and to document what you've been working on for forever and put it in, make your claim. And and I think that most people don't understand how important herbariums are, because they're like the basis of all plant science, and that didn't exist for cannabis. So the fact that we're doing it is so important and cool. It's just like the perfect marriage of science and art, I think. And I
think there's a lot of people that would be
cool.
really interested to follow along. Are you on social media at all? Is there a way where people can kind of follow you along
there's
and
both
see what
a
you guys are doing?
Leafworks and a Candor Instagram. People seem to gravitate towards Instagram.
Sure.
I'm the social media recluse of the group, so Charlotte is in charge of social media.
Nice. I've been busy, so it's kind of.
But we're working on. We really do a lot of work and don't do a lot of social play, but we should do more of that, because I do think people are interested in it. The herbarium is very special. This is the only plant that doesn't have really a dedicated herbarium until we started the herbarium. So this herbarium was also started in 2015, but was officially turned into it's a California mutual benefit nonprofit. So it is a nonprofit. It can't quite reach for federal status until some of the kinks in that system get worked out, but we are established in California as a nonprofit, and it's just a fundamental resource. So it's like that whole jar issue, right? So if I'm going to do a test about lavender, there's maybe 28 cultivars of lavender that are important commercially and have different values and are very unique plants, and they're small profiles and all of those things, and they're definitely they're established. So the first thing I'm going to do as a geneticist is not say, I'm going to tell you what this lavender is. I'm going to say, no, I'm going to go to an herbarium. I'm going to get herbarium samples of those different lavenders and take DNA from them. So then I can find out, what makes one different from another, and that's how I'm going to tell them apart. And that resource is just doesn't exist for cannabis. And so in that moment, it became very clear, if we want to do good science, we have to start a herbarium, because you can't do one without the other. And so it was kind of like, we're going to start a company and a nonprofit.
Wow.
But it was an important thing to do.
It's a noble cause.
In order to, do the work correctly. And then it's this resource that the community can reach towards when they have issues. So, there's been there's been all kinds of different ways that herbarium has been used already. So we had one instance where a farmer was growing a very special plant that they had bred, has been in their family 2 or 3 generations, and they were selling it through a co-op. And sort of the same thing as the grape example earlier. This person produced 100 pounds of this plant, and then when you look at the registers at the co-op, they've sold 150 pounds of that plant.
So where'd it come from?
So this person's pissed because they're like, first of all, you got to pay me for the 50 pounds you sold, even though I didn't produce them. And you're labeling my stuff as my stuff, but something else is in that jar, because you're able to sell it.
And you can damage
that's like a double
brand.
issue,
you can have damage in the brand.
Yep, absolutely. And so that's one of the ways that the herbarium has been referenced.
Well, that's another way prohibition has set us back generations, because in a different place in a different time, and today even, there were people that would be rated for having just the samples in the herbarium.
Oh, totally.
And do a lifetime in prison, or 10, 20 years. And so I think that that's a really important distinction to make, is that our federal prohibition has set back our medical and genetic science on cannabis at least 100 years.
Yeah. Mm-hmm. We did some work with the Missouri Botanical Garden, and I got to go there and do a lot of sampling, which is the 3rd largest herbarium in the world. It's one of the...
It's in the top 5.
It's huge. I mean, it's like 5 levels, football fields long, like huge, just rows and rows of herbarium samples. And we found the cannabis section, and there was like 5.
Wow.
And they also had poison, specimen poisoned. And so, what we have here is so important, and it's just so cool. Like, I love the herbarium so much. I can talk about it forever.
Where was that? In Missouri?
Missouri, yeah. They have a couple, there's herbarium all over. I think Kew Gardens is another big one.
The Kew is one of them. It might be the largest herbarium. And New York Botanical Garden is also is a very prominent botanical garden. The nice thing is
I grew up going there as a kid.
Yeah, the New York Botanical Garden has an excellent collection of conifers. Like, if you want to study conifers, that's the herbarium that holds the most samples of conifers.
Cool.
Right? So different herbariums have different specialties. They'll take plants, all kinds of plants.
Yeah.
Like Mobot has
every kind.
Thousands of, probably hundreds of thousands of different species of plants.
Wow.
Yeah.
But we're doing it we're doing some other tests for some other species, and so we're going there to get vouchers so that we can make references. So not cannabis, outside of cannabis.
But that's what you do.
It's
you
just
have to go to
how it
the
works.
specimens first. You have to go to their herbarium.
Wow.
That
super
trip was
fun.
cool. I also got to see some samples from Charles Darwin they had there. And so
Amazing.
You could theoretically go and take a little piece of that plant and sequence or extract DNA from it. So,
Yeah, or chemistry.
Yeah,
You do
chemistry.
like basic chemistry, not the volatiles, but the more stable compounds. So they're really useful. And one of the interesting things, the department that I did my PhD in is a genetics department that's had a lot of influence on the speciation, literature within evolutionary biology. And it's cool because in the plants, sometimes you'll have these things where you're like, it doesn't quite add up. The genetics makes 2 groups, but we're lumping them as 1 group. And then now we find there's this weird 3rd group, and you're like, what's going on? And then you go back to the herbarium specimens and you really look at them, and then all of a sudden you start to notice, like, oh, we got 2 species here that we were lumping as 1, or, oh, this isn't a 3rd species, it's a hybrid. But in the beginning you're calling them all the same thing, but then as you start to dig in, you realize, oh, they're not all the same thing, and so you go back to that resource. It's just a tool, right? And you can use it for all kinds of things. And cryptic species have been elucidated through using herbaria, hybridization, all kinds of fun stuff. You can go back and look at pathogen history. So it's really cool. Like, we've thought about, our herbarium is not so old, right? It's 2015, but we have some accessions that are older, like the Missouri Botanical Gardens. Mm-hmm. And you could say, do those herbarium specimens have HLV in them?
From the 1800s?
When do you start to see these things pop up? Or where do you start to see these things pop up? And so as it grows, the utility becomes more and more deeper. Like, there's more and more things you can do.
Well, it allows us to take the modern science and the modern questions and look back to a time when those questions weren't being asked and reference it to connect the dots from past to future. And that's really fascinating.
It's so cool.
Is that when they started to collect? Was in the 1800s?
Oh, no.
You have before then. Yeah, I think the
1500s there were some.
Yeah, and you have the first herbaria were really used by herbalists to teach their students what plants to collect. And so you have hundreds, actually thousands of years ago, you have herbaria.
Oh, wow.
I wonder how much has been lost through, the collapse of civilizations through the generations too, and how much of that was captured in, say, Rome and the burning and looting of Rome, and if there were samples that,
Yeah, that's interesting.
Yeah,
I'm
it just
gonna go down
gets
a
my
YouTube
mind.
rabbit hole later.
Yeah, it just gets my mind tumbling. You said it best. There is way too much to cover in one episode here.
I love scientists, though. You guys have been great.
So fun. Yeah, there's just so many fun things to learn, and that is what's really fun about cannabis as a whole. There's so much to learn,
and so many people asking the questions for different reasons.
Yeah, and it's a really engaged citizen science community. So you have a lot of scientists that are in the community that are, they might be physicists or chemists or biologists or geneticists, and they just grow cannabis or have been growing cannabis. So you have a lot of just, scientists in community. But then you have a lot of citizen scientists who are just super curious, very intelligent, question seekers, and that is one of the things I love about this community is how I mean, sometimes it can go off into some weird tangents, but the questioning and the exploring of ideas is amazing, and I think important, and happens a lot in this
It's
community.
the excitement of something new. I mean, obviously, even as for growers, I mean, it's something that people might have been doing for 20 or 30 years, but in a very basic sense. You got clones from your buddy, and you use the nutrients that your friends use and have success with, and then obviously, as we're getting more and more into it, everybody, if anybody's looking to be a successful grower at this stage of the game, like, you really got to start to break it down and think about things and why you do things and why they work. And so, yeah, it's been interesting to see the entire growing community elevate and become more educated. And this is the thing when you go to places like MJ Biz or Emerald Cup and you get in these conversations, and it's like, wow, everybody's become so intelligent with these conversations that there's an excitement there, not just because it's the livelihood that a lot of us are relying on, but it's new and fascinating. It's something that it's like the excitement of diving into something new.
Well, how many new things are really out there in the natural world, and being able to ask these questions and look at it through a new modern lens, I think really gets people excited. People that have been asking these questions without access to real answers for a long time are now diving headlong. They're like, we can get answers. Let's do this as a community. Let's grow it and understand how to make better medicine and what makes better medicine.
Yeah.
That's what I was saying about, being scientists. I'm like excited for you guys because I know that's like the scientist thing, right? When you get into something new, you hear about somebody talking about space that studies space, and they're finding a new, something new in space, right? And they're like the level of excitement, like Neil deGrasse. I love that guy. Like, he's freaking passionate. He's just excited. I don't know what the fuck he's talking about, but I'm like, this dude is just hilarious. He's got this big smile on, talking about the black hole and the infinite of the universe, and that's what I love that part about science. It's like explaining how waves travel through a wire or whatever. It's like, this is something that you guys are obviously really passionate about, and you can feel the excitement through your words, and it's been really awesome.
Yeah, it's so fun.
Yeah, we have a lot of fun.
And that's really just amazing. I appreciate you all taking the time, letting us come in and crash your world today. Obviously, you have a lifetime's worth of work ahead of you, so we don't want to keep you from what you have to accomplish today, because it's important work that you're doing. But I do want to say thank you so much. My mind is blown, and I want to ask 1,000 other questions, but if we start going down this rabbit hole, we'll be here all day. So
big ups to Charlotte too. I didn't even know she was getting on a podcast today. It's awesome. Like a rock star.
Another day.
Pinch hitter rolling out there, ready to go.
What are we doing? Bet. All right, let's do it.
Let me get a bagel first.
Right?
Yeah, thanks for letting me eat some lunch first.
Yeah.
That's awesome.
Well, thank you all at home for tuning in, checking us out. Eleanor and Charlotte at Leafworks. You can find them online at leafworks.com.
Leafworks DNA on Instagram, Candor Herbarium on Instagram, leafworksdna.com.
Just leafworks.com.
Leafworks.com.
Yeah.
Shout out to Karen too.
Yeah.
Yeah, shout out to Karen, my co-founder and work wife, who's not in the office today.
Yeah.
Well, thanks again for joining us. I'm Michael Beck.
I'm Rick Elliott.
And you're listening to Royal Grown Radio. We'll see you next time.